we seem to have a handle on the molecular basis for the major neurodegenerative diseases, which are really misfolded proteins in certain conformations. What is Amprion's unique contribution to this area, and why do we believe, those of us at Amprion and a number of scientists elsewhere, that Amprion's technology can be game-changing for the diagnosis and treatment of these diseases? So, to recap, the neurodegenerative diseases, Alzheimer's, Parkinson's, and the tauopathies, seem to be caused by misfolding of one or more proteins. The major proteins that we focused on for neurodegenerative diseases are Abeta and tau, both of which are involved in Alzheimer's disease, and Synuclein, which is predominantly involved in Parkinson's disease. So again, to recap, Abeta, normal functions, important protein. However, under certain circumstances, it can misfold into two different shapes. One of which leads to very, very large clumps, which are one of the microscopic hallmarks of the disease. Second, which leads to small soluble aggregates, which cannot be seen under the microscope, but really appear to be the part of the protein that's actually causing the disease. They are small, they can move from cell to cell, and this is how, if misfolding starts in one cell, it can spread to involve the entire brain. Tau also forms two different types of misfolded structures, very large structures called tangles, and small aggregates, which are invisible under the microscope, but yet seem to be involved in the progression of the disease, and function as the actual agent of progression. So what Amprion can do is uniquely detect these small soluble aggregates. Existing technologies can detect the normal forms, but for every misfolded small aggregate there may be 1,000 or 100,000 normals. This can be detected. The large aggregates have been detected for over 100 years under the microscope, but there has not been a reliable, very sensitive technology for finding the small soluble aggregates that are carrying the disease.